Now let us talk. Let us in this total discussion of mine. We are first thing I'm going to talk about the main characteristics of spirochetes. Among where we are going to see the different types of spirochetes and what are their general features of their cell. And the second thing we are going to talk about the human pathogenic spirochetes. Among them we are talking about the morphology, classification and physiology. We are not going to talk about diagnosis and treatment. We are just going go up to the epidemiology and till there. Okay, now the main goal of our discussion is to learn about the spirochetes, okay, not learn about the diseases. Now there are three types of spirochetes, as we know, as we are going to talk. One is Treponemus, another one is a Borrelia species. Treponemus or treponemes cause disease which is called the gonorrhea, as we know. No, Treponema causes the disease, as we know, syphilis. And Borrelia species cause the disease relapsing fever and also Lyme disease. And Leptospira causes different types sets of disease we are going to see later. Now let's first talk about the general feature of a spirochete. Now as we can see in this picture, the spirochetes are really thin. They are helical in nature. So this is the characteristic shape of a spirochete. So whenever we are talking about a bacteria, as we know, first thing that come to our mind when you look bacteria through the objective lens, through the eyepiece of a microscope, we find something like that. Sometimes like this round shape. But in this case, the first thing you can see when you look through the microscope, we see a spiral structure like that. And this coil structure is a very important characteristic shape of the spirochetes. So whenever we are looking at this kind of structure, and also this bacteria is motile, they can be transferred from one place to another place by using the screw-like motion. We can say the bacteria we are watching is a type of spirochetes. And these bacteria are gram negative, that means they are having three layers. We are having the inner cell membrane layer, then we have the peptidoglycan layer. Suppose this layer is a peptidoglycan, then up a layer of the peptidoglycan, there is a layer outside the peptidoglycan which is the external cell layer. Though the peptidoglycan layer is really thin in this case, but still peptidoglycan layer can be found in those cases, and they are having the flagella for their motility purposes. But the important thing about them, the flagella spirochetes provide, is having the differential structure, different type of structure than the flagella what we can see in normal bacteria. Now as we know that normal bacteria produce flagella from the inner cell layer, and it just go through the passes through the inner cell membrane, then passes through the peptidoglycan layer, and then find the outer membrane and just released outside. So if we think about so let me erase all these inks. Okay, and suppose this is a bacteria, so this is one of our bacteria, and this bacteria possesses a flagella. The flagella will look like this. If this is a bacterial cell and the flagella come out from the bacterial cell, it will be outside it is going to face the environment like that. But if we think about the spirochetes, what will happen if we look at the spirochete structure, look at this. So this is the spirochete structure. It's a coiling structure like that. And let me change the color. So suppose this is the coiling structure, and what happens in case of spirochete, this is the cell of spirochete. And so from this cell of spirochete, if I cross if I say cross make a cross section of the spirochete, what we can see some region where also this flagella is coming out. That means, suppose this is a straight chain, this spirochete. I am not coiling this up now. Now what happens, they produces flagella from their inner cell membrane. So suppose this is the inner cell membrane, so I am this is the inner cell membrane and this is the outer membrane. So I am making it really big for your understanding purposes, but not this is not the case. So here is also we are having, so this is the cytoplasmic portion of this spirochete, and these are the outer membrane and these are the inner membrane of spirochetes. So this huge region which I draw in this picture, so actually they are not huge, but I draw them because of the understanding purposes. Now these huge gaps are the periplasmic space, as we can know. Now the flagella of this spirochetes usually generates from a region like that. So let me change the color again. Let me take the red color. Now here we have the flagella coming out from the inner cell membrane, and it is just released into the periplasmic space, periplasmic space, instead of go outside the cell. So as we can see in case of traditional bacteria, that the flagella is generating from inside the cell and it faces the outside the environment. But in case of the spirochetes, the origin of the flagella remains the same, but its ending point is different. It just reside in the periplasmic space. So these spirochetes are having a huge periplasmic space. In that periplasmic space, they are holding their flagella, and that's why, as we can see in this case, so if we think this is the flagella will go in this direction like that. So when the flagella start to move and they just wrap around themselves around the flagella, that they can move in a screw like movement. As we know that if we take a screw and if we tight them, if we make tighter, and it will finally go into some direction. That's how this kind of spirochetes start to move, because they just make themselves like a screw and they can move from one place to another place. Okay, now we are going to talk about three types of pathogenic spirochetes. One is treponema, another one is borrelia, and leptospira. Now in among all these three types of spirochetes, what we see in common is this kind of twisting structure of their cell, this kind of spiral structure of their cell, as well as the presence of this periplasmic flagella, which is called the axial flagella. Okay, now this is we can see in common, but what will vary from treponema to borrelia and leptospira is that the presence of flagella and the number of flagella that they possess. So maybe in case of treponema it is only 3 to 4 flagellas, in case of leptospira it is only 2, or in case of borrelia it is more than 5 flagella we can find. Okay, but the presence of flagella and the arrangement of their cell along with flagella remains the same in all these cases. Now if we go through the classification scheme, then we can see in the treponema pallidum, this treponema pallidum is a causative agent of syphilis, as we know. Now syphilis is a venereal disease. This is also among the sexually transmitted diseases, as we know, and this syphilis disease is really dangerous, because this disease usually do not set for just spontaneously, because it needs time, it needs the incubation period to hold on your body. So there are first or primary situations when they found the environment feasible for them, they start to grow, and finally it will produce some symptoms or sometimes no symptoms. We call the stage is a primary stage of syphilis. It just begin like skin lesions, then it finally grows on and on. It will produces much more lesions and cramps, and finally it will go to the stage of secondary syphilis. In the secondary syphilis stage, what happens, sometimes fever, nausea, flu-like symptoms. So often people misunderstood this kind of symptoms as flu, so they usually do not treat in those situations. But this lack in their knowledge and treatment, this Treponema pallidum start to grow again, and finally go through the final stages like tertiary and quaternary stage, when they start to invade the tissues and just degrade those tissues up. Sometimes they can attack in the neural tissues, and it will create the neurosyphilis. That is also very dangerous and destructing disease. It also can be transmitted via the sexual contacts, and that's why safe sex is a must thing for preventing this kind of sexually transmitted diseases. Okay. Now, if we look for the morphology and structure of this Treponema strain, and we can find this organism is so thin, tightly coiled with the straight end, not seen by the stained smear. So we cannot see them by usually look at the stained smear, or by looking at the using the light microscope, by the normal compound microscope. We need to visualize these things only under the dark field microscopy. So we need to make the field darker, then you can see this kind of spirochetes on this microscope. Now if you do not know what is dark field microscopy, please go in my website and look for the dark field microscopy tutorial. Now let's move on. Now we can also find this structure using the fluorescent microscopy. So we can tag the fluorescent dye with this spirochete cell, and then we can visualize those fluorescence which is coming out from that spirochetes, and we can look at them. Okay. Now this another important thing about this Treponema is that they cannot be grown in vitro conditions, so we need to provide them the original cellular environment like that rabbit epithelial cells, and they need a huge amount of replication time, or what you can say incubation period, about 30 hours or more than 30 hours to grow and reproduce. Okay. And most of them are facultative anaerobes, and that means they do not need oxygen for their growth purposes. Okay. So sorry, yeah, right. So they do not need oxygen for their growth purposes, and they can if the oxygen is present, that's fine. They can use glucose, and they can use glucose via oxidative manner. Okay. So that is another important biochemical feature of their cell. And as we know, they are having the three periplasmic flagella, and these flagellas are inserted at each of the cell end or cellular ends, as you can see, under the outer membrane envelope. Now if we look at the picture, it will be more clear. Then now look at this. This is a clearer picture, as you can see. Now this is the actual body of spirochetes, which is denoting here with yellow color, and this is the pink color portion is the outer cell membrane, and inside, in between this yellow and pink color region is which is the periplasmic space. This periplasmic space is containing this long flagella, as you can see, which is just attaching both of their ends. Now this is really important for their movement, because if they produces flagella from one end, it will go to it will not attach to the and it is not attached to the outer the other end, then this type of coiling structure cannot be achieved. So they need to achieve this coiling structure for their movement. They need to achieve this screw-like structure for their movement, and for that purposes they need to attach both of their ends with this flagella. Okay. and with this periplasmic flagella. Now we can look here. This is the cytoplasmic membrane, as you can see here, and here is the peptidoglycan layer, which is really thinner, but the periplasmic space is much more profound in this case. And here is the flagella is coming out, and it is just going to touch to the outer membrane region, to some region, and it is the generation point. Like this is the point of generation, this is another point of generation. This picture is zoomed here for these purposes, and it in turn is going to attach with the other end of this spirochete. Okay, so this is the morphology of this flagellar assembly. Now what will vary to the Borrelia or the other types of spirochetes, that you can see the number of flagella, but the arrangement of the flagella and the morphology will remain the same. Now if we look at the treponema, and then the flagellar components or the structural proteins are the same like bacteria. So we have a phloem, phloem, and we have the MS ring, we have motor, we have rotor, and all these things together. Now if we look at the treponema pallidum through the dark field microscopy, we can obtain a slide specimen like that. Now you can see treponema pallidum. These are the screw-like structure, as you can see. So this is the characteristic structure of spirochetes. You can see, you can just identify by looking at it very easily. Now if we look at this structure using fluorescence microscopy, actually it is much more attractive picture, because fluorescence is always very attractive indeed, because we are using two different color in this case to visualize that. Now if we are using them for different purposes, for staining purposes, it cannot be stained in normal way. More especially this treponema cannot be visible using normal stain. That's why you need to go through either of these techniques to visualize these bacteria. Now the pathogenesis, as we know, they have the virulence factors. So what are the virulence factors for them? It is the first one is the outer cell